So this general introduction is good to make some reflections, but does not help us to say, okay, I can do this with your data. Now, the second part of the talk, I will turn from general to very specific. What can we do with, for instance, those types of data sets? And I will mainly talk of an application we've been doing using multivariate statistical methods to make this combination. This is something we could do with your data. Specifically, I am going to show how some general ideas and some applications of how using a multivariate technique, which is called multifactorial analysis, to combine and visualize multi-way data sets. After this, I will give some brief ideas on other possibilities, but this will be the widest application. Okay, why this technique? Well, there are two reasons. First, this is one of the things we know how to do. This is a statistical technique. And the second is that it is very common. See, said 5 years ago, it was very rare that a researcher had nothing more than one data set. For instance, after spending a considerable quantity of money, he might have some microarrays for their individuals. Eventually, we also might have clinical variables. That also already was two data sets. But as the price has gone down and the complexity of researches has increased, let's say the data sets or the different types of information that are available for each individual have increased. For instance, this is a cancer example which is based on two public data sets related with gliomas, which are brain tumors. And in this case, these researchers had collected, by one side, microarrays. They had, I don't remember, it's about 43 different microarrays of 4 different types of gliomas. And by the other side, using CGA arrays, they had collected information about copy number variation. So, and these they had for every individual. This is one restriction from the approach we are considering, although we are looking for ways to overpass this. Which is the restriction is that in principle we are happy when we have for each individual the different types of data. Had we, for instance, transcriptome data for for a set of individuals and CGH data for another set of individuals, wouldn't be able to apply this or to use this approach. Okay. Well, a second example that we have faced, the idea of showing these examples is that it is very easy to find somebody explaining my data consists of this. For instance, this is a real case we are working with some researchers, and they are investigating insulin resistance associated with diabetes, and they have collected up to now 3. Although indeed it is 4 types of data sets. By one side they have clinical variables, some general and some that they relate to insulin resistance. By another side they have collected what what they informally call DNA bands, which are, it's a special type of, or a particular type of DNA electrophoresis that is, can be identified. For instance, it is used in metagenomic, in a kind of primitive metagenomic experiments to distinguish the microbiota composition in different situations. And by the other side, they have collected, they have done microarrays. Indeed, these researchers keep on working with this data. And for instance, now they are doing microRNAs and ultra-sequencing. So the number of data sets grows. Then the question is, for instance, in that case, these researchers came to us only with these 2 data sets, and they asked us, what can we do with this? And sometimes later they started, now we have microarrays, now we have that, and now. And the question is always the same. Are we able to improve the explanations we give when we add more data sets? Okay. Combining data is not a new problem. Indeed, it is rather old in fields such as chemometry or ecology. In ecology, for instance, they try to combine, they have data sets on species and ecosystems and chemical variables. This is multi-way data sets is a very common issue. And in consequence, there have been developed many different techniques, especially multivariate techniques, to work with this data. We did some trials with some investigations about which may be a good technique to work with it, and we decided that multiple factor analysis, which is not the same that factorial analysis, by the way, could be an interesting option because it could be adapted specifically to the nature of these data sets. Okay? Now, I will give a brief explanation, a non-technical explanation of what this technique is. Indeed, multi-factor analysis defines itself as a multivariate technique which is adequate to analyze heterogeneous data sets, either quantitative or categorical or mixed variables. So, what is multifactor analysis is an extension of one of the most common used and most well-known techniques used in biology and other fields, which is principal components analysis. Principal component analysis, which is also very not only used but also abused, is a technique which help us to find, let us say, that what it does, it transforms the original data in such a way that it creates new variables which are called principal components, which have the property that these variables are by one side orthogonal or uncorrelated, each one of the other. So each of them explains a different thing, and also each of them explains more than the next one. So if we start with a set, let's say, of 3, 4, 5, 20 variables, we can transform it into their principal components, but we probably won't need the 3, 5, 20 principal components, because perhaps using the first, the second, the third one, we will be able to explain the most part of its variability. So, given this property, principal PCA has been very used to do many different things. I will not, of course, go into it, but for instance, the main issue for it is used is to visualize data in low dimensions, because, as I said, we may not need all the dimensions. And given for this reason, it's also called a dimension reduction technique. It has an advantage, and a very important advantage, and it allows to visualize simultaneously or in the same coordinates the variables and the individuals. And so you may put them together and create visual associations. And this has made that it is used for many different purposes, from quality control, because you can plot and see if an individual is an outlier, for classification, for detecting structure in the data, and so on. Okay, but this is principal component analysis, and the idea is that it is a technique that works with a one multivariate data set, let's say, many variables with many individuals from many samples from one type. Now, multi-factor analysis or multiple factor analysis is essentially to extend this to different data sets. Let's say I could be able to do PCA with using, for instance, the expression values, the DGA values, the clinical variables. Then what does MFA is simply to perform separate PCAs, to perform separate principal component analysis, and then to weight them. So that is, to combine them using well, the eigenvalues, which is a technical term I'm not going to define now, in order to obtain a global PCA that is essentially a PCA performed with all the data. That's we can see it as making a PCA combining all the information. Okay? Well, that's the technical idea, and this is more difficult to explain than to do. And the good thing is that it offers us, by one side, it is very easy to yield graphical interpretations, which are graphical visualizations that are relatively easy to interpret. I will give you some examples now. And the most interesting thing from a biological point of view is that multiple factor analysis, in the same way that PCA can be done using what we call essential or instrumental variables, that is the variables that we use to create the principal components, but we can add new variables onto it, that is what we call supplementary variables. And these supplementary variables that we can put on the picture to visualize, for instance, can be biological knowledge. If I have, for instance, information related with gene sets that are affected by given processes, I can combine with these data sets. So not only can I combine different types of data sets, but I can add different new information or completely different type of information into the visualization. Altogether, can give a global picture that is probably more explanatory than simple ones. That is more or less the idea. And an important thing that before anybody asks the question, how do I do it? It is very easy because there exists software available for this in the R system that allows, without having to program, without having to go deep into the concepts, to use the technique. So it is a technique which is powerful and at the same time relatively simple to use. Okay, well then given the situation, for instance, I am now going, originally I had the insulin resistance example prepared, but it is yet unpublished results, so the researchers have preferred that I hide them. And I, for instance, just show some of the things we can do using the gliomas examples. For instance, one of the, as I said, we can use as I said, the principal components to represent either the individuals or the variables. Representing the individuals helps us find if there is a structure in the data. For instance, in this case, we have 4 types of gliomas. I am not able, I think it's glioblastoma, this is astrocytoma, and the other omas, I don't remember. But it is the interpretation that we can do always with this type of pictures is that we have to look at the components and to see if there are different groups that appear associated with the different axes. In this case, we can see, for instance, that the glioblastomas are in one side of this dimension, and the other types are on the other. So the first dimension, we can consider that it separates the glioblastomas from the other. And the second dimension, where we can consider that can be associated with the different types of other tumors, because we can see that, of course, one could argue, I would argue myself, that if the points were not colored, this would be much more difficult. But if we look at the centers of each group, it is relatively clear, for instance, that this group has a positive value and it's to have a negative value. So it is relatively easy to see this association. It is very common in this type of pictures to think that we are somehow forcing the interpretation, that when one has seen many of these figures, one realizes that some of them we have to help the interpretation and some of them are very clear. So, for instance, we can look at the variables, and looking at the variables, we can do, the idea is that the longer each arrow is for one variable, in this case one gene, and the longer the arrow, the more explanatory or the more associated is this arrow with this component. So we have the red arrows are associated with this component, and these are the top correlated genes, and the green arrows are associated with these components. So these are, for instance, the genes that explain the difference between glioblastoma and the others. And what is interesting of these pictures is to remember that although this is the same that we would do with the PCA, we are doing with all the data. So we are helping, we are obtaining a better interpretation using we're obtaining a better interpretation using the different data sets, although we don't see them here. Perhaps it's not that important if we are not specialists in explaining with a high detail each of the pictures. But in this case, for instance, what it is shown, again, is that for instance, that the second component is most associated with the CGA data, because if we look at these are the positive and the only positive values, we see this is the expression values. We see that it has a very low value with this first component. So whereas the CGA has a very high score. So it gives what it is saying here is that CGA is explaining something different than what is being explained by expression values. Well, we can also ask ourselves what would happen if we did separately, if we had not done this multiple factor analysis, but we had done separate PCAs, and this is what can be seen as this picture that says partial access. Again, for instance, here we can see that the CGA has two dimensions, because we see two arrows which also reach the circle, whereas expression has only one big axis. The number of dimensions, of explanatory dimensions associated with CGA is 2 instead of the number of explanatory dimensions of expression, which is only 1. There are some things that are explained by expression values, but more things are explained by CGA. Okay, and last, how can we combine? How can we combine, for instance, biological knowledge with these explanations? To do this, what we do is we take, for instance, the genes that have been differentially expressed. We do a gene set analysis that allows us to detect sets of genes which are particularly associated with, for instance, a given biological process. And then we can treat each of these gene sets as supplementary variables. And once we have a supplementary variable, we can plot it jointly with the quantitative expression and the CGA's variables. That would be a representation of what does this mean. What this means is that we have each set of genes. Each set of genes is taken separate by one side together. Saying we don't take one gene, we take a gene set, or a module, in other terms, and we don't use this information to perform the PCA, but to project it into the coordinates. Okay. Well, the way we project it is a bit tricky, so I don't go into it. But what can be done after the projection is to look, for instance, at the gene terms, the Go terms that appear more associated either with one or the other dimension, which allows us to give terms which are more associated, for instance, with expression, or with more than with expression, with things that are associated with each component. For instance, in this case, the researchers were able to identify different types, a series of biological processes that were either associated with more with gene expression or with CGH. Okay, so altogether, what we can see with this technique is that we are able to, by one side, to combine different types of information, and not only to integrate, let's say, the information, but also to add biological information on it. Altogether, it can help us to do things that we would expect from these situations, like highlighting associations too vague in a normal situations to be detected. Okay? Well, of course, this has limitations. By one side, this is an exploratory technique, so we're not going to have any p-value, and many people have said about it. And also, it is difficult to deal with missings. That is, we expect that we have everything for every variable, for every individual. Of course, it is good for a researcher who may start thinking on how to deal with missing values. We started thinking of it, but the people who did the method also did, and they have also developed a variant on how to do this with missings. Okay, so it's some of the limitations are relatively easy to overcome. Okay, that's the nucleus of the presentation, a way to combine or integrate different types of data. Before I finish, I would like simply to comment that data integration, analytical data integration, can be performed also at other levels, and for instance, or for other type of problems. And one of the problems people is very concerned with in biomedical environments, for instance, is class prediction. I can say that many people who come to talk with us comes to say that they would like to obtain a predictor, a signature, a biomarker, and so on. So the question is, can data integration help us to improve the obtention of predictors of classifications? I'm afraid that this picture was too small to put in the slide, but it was simply to remember that although building predictors is one of the things that people is most interested in, this is also one of the most difficult problems to solve successfully. It is very easy to realize that if building predictors and finding signatures was that easy, there would be many diagnostic kits based on microarrays. And from thousands of experiments that have been done in the last decade, approximately there are not, the number of diagnostic kits based on microarrays is less than 5. So it's clear that building a good predictor is a challenging and difficult task. So the question is, can we profit of data integration to do things better? Okay, probably we can, because by one side, if we, for instance, are able to combine heterogeneous data, we are going to have a richer information that may give us, that may make more things to be used in the obtention of the predictor. By the other side, if we are simply combine different data sets, we are going to have a greater quantity of information, or simply having different data sets can help us to make one of the important things that is having independent data sets for building and for validating the predictors. Okay, there have been suggested many approaches, and each of them would require a talk. So I just would say, for instance, this is a very interesting paper where these people used a kernel method. This kernel methods is a very wide field. But for instance, kernels are well known in the biomedical literature. For instance, in using supervector machines to build classifiers. So it could be seen as a variant of this. And they use a kernel method to combine different heterogeneous data sets to build a predictor. Why is this done? So this may be easily justified because kernels have, in given situations, the idea of combining, of adding different kernels is very straightforward. So they profit of this approach to build a kernel for each different type of data set, merging them into a new one, and using this for building the predictor. For instance, a completely different approach is to use a Bayesian approach, where we combine, we have prior information, we have observed data, and we use the Bayes theorem to make a posterior distribution on which we rely the inference and the models. And in this case, what these people have done is to use the different data sets as a prior, and then use a Bayesian approach to combine some of these data sets taken as a prior with another data set. Okay? And last but not least, this is a company that offers a data integrated approach to biomarker. And for instance, they take a completely different approach. What they do is they rely on public data sets, which are not heterogeneous, but are that refer to the same type of data, and they make in the style of the meta-analysis combination, they let's say refine their predictors using more data. Okay, so of course I don't hope that nobody has understood the details of this, but the idea would say that we can tackle many different approaches to build good predictors by integrating data. Okay, so in summary, what is the idea? The idea by one side is that having multiple data sets of different types, it is unavoidable that we are interested in being able to combine them to obtain better explanations. This is one side, one of the aspects. Of course, this requires different efforts to be done. So there is as much a bioinformatics, a computational effort, a mathematical modeling effort, and so on. And of course, there is no unified theory. We often joke by saying that we are looking for the integrator. That is a method that when people come to us, we can say, okay, your data will go through our algorithm that will integrate and will give you a clean solution. But I think that we still have to wait. But because up to the moment, one of the things that we have learned is that we have to treat each of problems separately, but we still think that we can find a unified theory, okay? Okay, and just to finish, I want to thank my research group at the University of Barcelona, my colleagues that support, and support means many things in my case, at the Unitat d'Estadística i Bioinformàtica and the Unitat de Serveis Científico-Tècnics, who always support us. And thank you again for inviting me to give this talk. Okay. If you...
You know, in this system biology sort of models, which you have systems of equations with hundreds of variables. Do you know if this kind of PCA or this multi-factor analysis could be applied to that sort of to kind of do this model reduction and end up with something that it's more...
Well, I guess it could. I guess it could, because indeed it's nothing that an eigenvalues problem. So you may use, I think you probably know more than me that you may use transform a big system of differential equations into an eigenvalues problem, probably. Then I think that singular value decomposition and all those things are often applied. So probably it might be, there is already something done which uses this transformation, because essentially this is nothing more than a rotation to look for let's say, how do I say, prioritized, no, the optimum directions from where you can see the data well. So probably the answer is yes. And the second answer is I wouldn't know how to do it.
Anyone else? No? Okay, then let's thank Alice.
Okay, thank you.